Okay, so I think we're going to go ahead and get started. It's 1 o'clock on the dot. I hope everyone had wonderful lunch. Yeah, I guess kind of just to kick off with the question I kind of prompted you with this morning. And we just had lunch. So yeah, if you're here, I'd like to give me, what's your favorite food? If you were on death row and somebody was going to make you your last meal, what would that meal be? I think it's always kind of hard to answer this after you've had a meal. You're kind of like, I'm full, I'm good, but it's going to kill a little time before we get started. Oh, geez. Like everybody's like, carbs, man. We're going out. It's got to be carbs. It's great. Oh, popcorn. That's an interesting one. Oh, burritos. Yeah. Yep. Back when I was in graduate school, I think I actually dressed up like a burrito for Halloween. I was actually tin boil from pretty much here down, but it was pretty fun. Some gravy. Okay. So I hope everybody had a great lunch. We're just going to continue our conversation that we had before. Like I said, during my lecture, I keep my chat window open. So feel free to shoot me any questions. And if you are a person who doesn't feel comfortable shooting questions kind of publicly, just realize that you can also private messenger me as I'm lecturing as well. And that that message can be as simple as, hey, I missed that, can you go back? Or hey, slow down, or I have this other question. So just feel free to, again, at any point in time, reach out and kind of communicate as I'm lecturing. Okay, so let's go ahead and kind of continue where we left off. Actually, are you guys talking about biscuits and gravy?
Yes, yeah.
I've heard the Black Cat has really good biscuits and gravy, but yeah, I'm sure everybody kind of has their own favorite. Okay. So I guess something I wanted to go back to. So during our last class period, we had a discussion about L-DOPA and carbidopa. And I think that, Robert, you actually gave a really good explanation of this, and I think I might have convoluted it. So I actually just looked up this nice graph here that describes some of the metabolism of levodopa, and it undergoes extensive decarboxylation. So when it's given with the carbidopa, it actually blocks that decarboxylation in the peripheral nervous system, which allows for more availability of that levodopa to go to the brain, and at that point get broken down into the L-DOPA. So I just looked up this table during our lunch break here. I like this because it gives you kind of a little bit more information too, I think more pharmacology-based information. And I'll go ahead and save this version of this presentation. And so it'll be included in the one that gets updated after this lecture. So again, if you had any questions about this, I know somebody was like, hey, can you repeat that? And I kind of gave like my 2-sentence description, but I think this also kind of gives you a better mechanistic description and understanding of exactly what's going on here. And again, that's not necessarily the main focus of this particular lecture, but it does, I think, apply to pharmacology and other questions we'll be discussing a little bit later on, because we will be talking about, you know, different synthesis of neurotransmitters. So we left off talking about ion channels and receptor gating. And so we went briefly over this slide talking about the main ions that are that pass through ion channels in the brain. So things like sodium, potassium, and chloride. Again, I like this particular table right here, just because it does show you the concentrations of those, and those are the extracellular concentrations. Although it would be, I think, valuable to also see the intracellular concentrations, because it's the ratio between those two things that dictates driving force. But I think if you can remember that when I'm teaching this at a 100 level, I usually call a neuron a salty banana. So students remember that there's a bunch of sodium on the outside and that there's a bunch of potassium on the inside. I'm guessing you are all past that point of having to remember that the brain is a salty banana. But again, if you ever have that moment where you're like, oh, I can't remember what is what, sometimes it helps folks remember. So we talked about the voltage gating of sodium, potassium, calcium channels. Again, sodium, potassium, voltage gated sodium, potassium channels are very important in action potential generation. And we'll talk about when and how those particular channels are open, how long they're open for, and how that dictates action potential generation. Ultimately, everything kind of gets kicked off by a ligand gated sodium channel, and then that gets essentially converted into a voltage gated sodium channel. Once you've amassed enough sodium inside the cell, it's depolarized enough. And then as the action potential travels down the axon, you have voltage gated sodium channels that are essentially opening all along that axon. And then right behind them, to repolarize the cell, you have voltage gated potassium channels that are opening and allowing for the efflux of potassium from the cell, again, lowering the overall the cell current. And then once that action potential reaches the presynaptic kind of bulb, it's going to cause voltage gated calcium channels to open, and the influx of calcium is what's going to lead to the presynaptic release of neurotransmitter via the interaction of those calcium ions with those snare proteins that I mentioned last time. So again, that's a very quick synopsis of each one of those types of voltage gated channels. If you're following along and you're reading through the textbook, they'll kind of describe it to that level, but we'll see these players again. So I think this might have been this or the next one was the slide we left off on. So as much as it is important that we have cations influxing into the cell to cause depolarization, hyperpolarization must also occur. Again, you kind of have to have that push and pull for a signaling mechanism to work. And so chloride channels are generally how the cell becomes hyperpolarized or more negative. And so when the cell becomes more negative, you have influx of chloride ions. And so this dampens or inhibits neural transmission. As we said in the previous slide, we showed the table of the concentration of chloride. And again, you can see here the extracellular concentration of chloride is much higher than it is intracellularly, causing the driving force of chloride to be into the cell. So when these ion channels are open, chloride automatically wants to go into the cell. This decreases the internal voltage. We'll talk about the two ligand-gated ion channels, glycine and GABA, more extensively. I think we might get into it in this lecture. If not, we'll continue that conversation tomorrow. And then there's also other types of chloride channels that are present in the brain, things like the CLC chloride channels and the CFTR channels. In this particular section of the textbook, it goes through some non-selective channels. These non-selective channels that they mention are activated, a few of them by cyclic nucleotide bindings. They're expressed in different regions though. If we look at the CNG channels, those are in the sensory systems, things like the retina and olfactory neurons. And if this cyclic AMP or cyclic GMP binds to these, it's going to cause a non-selective influx of ions. It is composed of 2-alpha, 2-beta, and the HCN channels similarly bind to the same cyclic nucleotides, are also going to open with hyperpolarization and close with depolarization, so they're almost voltage gated in regard to. And then they're expressed in, like I said, in different regions. You see this in the heart and the CNS. The TRP channels are going to be important channels. They do a lot of different things. There's a whole diverse set of TRP channels, TRP channels throughout the CNS. Here you can see that there's 28 families and 6 receptor subfamilies. So there's a lot of different subtypes here. And you'll see kind of how those different families give rise to different specializations or different types of activations. They can be non-selective for cations such as calcium, magnesium, so divalent or monovalent cations. And then this is important for thermoregulation, osmoregulation. So this is an important set of channels that have been related to specific disease states like cancers and neurodegenerative diseases. And so because of that, it actually has been a source of research for possible therapeutics. And I also like to throw this particular figure on here because I think it gives you a good sense of how these different TRPV or TRPM channels, how the diverse set of them allows the body to take in different types of sensory information. So here you can see the temperature gradient along your x-axis here, and then the kind of the distribution or the temperatures at which these different types of channels would be activated and open. So here you can see kind of this receptors. These TRPA1 receptors are going to be activated at your much colder temperatures, and then your TRPV1, 2, and 3s are going to be activated via those warmer temperatures. So again, you've got a lot of different subfamilies here, and it aids in the specificity of the response for the body. Okay. In terms of characterizing ion channels, so as an electrophysiologist, this is something that really hones to me, because this is exactly the how I would characterize these types of ion channels, is by blocking them with specific agonists or antagonists. And so for sodium channels, if you're doing an experiment and you would like to block all the sodium channels in a cell, you can do that by applying tetrodotoxin. This is isolated from the pufferfish. It has a very high affinity for the sodium channels. Bacrotoxin also slows inactivation. It's from a frog. So it's interesting, as I mentioned in the first lecture, kind of the evolutionary source of how you have these very high affinity antagonists or channel blockers that can bind to these specific channels, and how they've kind of co-evolved. It's really interesting, and how the animals have probably created these as some type of way to deter predators. If you were going to, let's say, try to record spontaneous release of neurotransmitter from neurons, if you're looking at spontaneous release, you don't want action potentials causing release. And so when you're doing those types of experiments, you would apply tetrodotoxin, because it stops any types of action potentials firing in those neurons. And so that way you can observe just kind of a blank background, just looking at spontaneous release of neurotransmitters in the cell. But that's kind of, again, more kind of a research angle as to the applications of tetrodotoxin. Potassium channels obviously also have their own set of drugs in which you can use to block them. Apamine and carbatotoxin, again, they're coming from sources in the environment, and are going to be blocking the small calcium activated potassium channels. And calcium channels as well have their own set of toxins that are able to block those channels. You'll recognize these are all kind of considered toxins because they do cause adverse side effects. And again, all of these animals have developed these toxins as a type of defense mechanism. And this is kind of an abbreviated list of even the calcium channel toxins, because these are blocking specific calcium channels, and agotoxin and omega chronotoxin are, again, these are N and P type channels, and there are even further numbers of specific calcium channels that are present. Okay, so would you like to know the channel acronyms and what they stand for, like channel transient receptor potential, or just what they do? So are you talking about the CNG and the HCN and the TRP channels? Like the you just need to know kind of those abbreviated channel names. You don't necessarily need to know their extended ones, just as long as you know those abbreviated channel names, because I think that's more often how they are annotated in the literature, and then just make sure you know kind of where they're distributed and what their functions are. Does that answer your question? Okay, awesome. You can see kind of how seamless it is to kind of just answer questions as you guys have them. Okay, so anyone have a question before we move on? Okay. So we've gone over ion channels, and next we're going to go over receptors and transporters, but in a very general way, because once we've done like a general introduction to both of these things, we're going to go into the neurotransmitters and the neurotransmitter system. And for each neurotransmitter system, there are going to obviously be specific receptors and transporters that correlate to those. And so this is just a general introduction to those. So I'm guessing you have seen this slide in various forms multiple times in your scientific career, where you compared and contrasted ionotropic and metabotropic receptors. And I think we've even covered it earlier on in this particular course, but this is just, again, just a very general review. One reason I wanted to bring this up is because when we talk about neurotransmission, we're going to kind of characterize these two different types of receptors as either being fast or slow neurotransmission. So ionotropic receptors, as we say, are going to allow the passage of ions. They're going to be ligand-gated receptors. And so if a ligand binds to the receptor, it's going to open the receptor and an ion is going to flow through. They're composed of multiple subunits, and once that ligand does bind, it causes some type of conformational change and allows that pore to kind of open and the ion to flow through. This is a very fast process for these receptors. This is why it's important for fast neurotransmission. So right now, all the thoughts that you're having in your brain are being kind of mediated by these ionotropic receptors, and we'll talk specifically about what subtypes here in a few more slides. Metabotropic receptors, to contrast that, or G protein coupled receptors, those are 7 transmembrane receptors. They're coupled to secondary messenger systems. Those are more mediated through slow neurotransmission, and we'll talk about some of the G protein coupling I guess pairings when we get further into some of the specific neurotransmitters. So again, just kind of returning to this particular image that we've seen again and again and again, as you have release of neurotransmitter, it crosses the synaptic cleft and can bind to either G protein or ionotropic receptors. What's interesting to note is that a lot of neurotransmitter systems have both types of receptors. So there are multiple types of neurotransmitter systems where you have an ionotropic receptor that's present, and then a metabotropic receptor that's present that binds that one neurotransmitter. And then there are even subtypes within that particular neurotransmitter system, as we'll see in future examples. And so this is just an example of some ligand-gated ionotropic receptors. So here you have the nicotinic acetylcholine receptor, the GABA A receptor, the glycine receptor, and the AMPA receptor. And this is just giving some of the antagonists for those receptors, bunkerotoxin for the nicotinic receptor, picotoxin for the GABA A, strychnine for glycine, and flanotoxin for the AMPA receptor. And it kind of shows you here again what the effect it has on it. You'll notice that it's not necessarily just an antagonist. In some cases, you have an irreversible antagonist, something that blocks the channel, a competitive antagonist, another one that blocks the channel. What's going to kind of dictate how this is characterized, an irreversible antagonist, not shockingly, is going to bind to that receptor and not have an off. The block of the channel is going to be different than a competitive antagonist, because a competitive antagonist is going to compete for the binding site. But these are other pharmacological terms we'll probably talk about a little bit later. And you can see here kind of the where these toxins are endogenously expressed in the environment. So like I said, it's a very general introduction to receptors. Again, a 30,000 foot view. We'll go into a lot of different receptors when we talk about the neurotransmitter systems. Okay, so the next thing we'll be talking about are the transporter systems. So ion channels are going to cause essentially neurotransmitter release. Receptor, once the neurotransmitter is released, it diffuses across the synaptic cleft. It binds to postsynaptic receptors, and kind of the last step in that process is going to be the cleanup of that neurotransmitter that gets released so that's not binding to those receptors. And so you basically need a system in which you either have enzymatic degradation of the neurotransmitter, or you have transporters that are expressed on the surface of either the neurons or the glial cells that are going to uptake that excess neurotransmitter and stop it from excess signaling within that synaptic cleft. And so Dr. Parker talked previously about the how organophosphates bound to acetylcholinesterases irreversibly. That's, again, one type of way in which synaptic signaling is terminated. Another type is through transport, and so we're going to talk a little bit more about transporters here. So it's just kind of a contrast of that. So transporters, again, sorry, I'll come back to this question. So as transporters assist in termination of neurotransmission, there are some, again, other ways, as I just mentioned, of the enzymatic functions. So they're going to be expressed on both neurons and astrocytes. You're going to see probably a larger proportion of them expressed on astrocytes, but then also in different regions of the neurons. Often those transporters won't necessarily be expressed right in the synaptic cleft. Sometimes they'll be expressed perisynaptically, which is kind of like the outside of the synaptic cleft. If your synaptic cleft is here, kind of perisynaptically is outside of that, just subtly. So if you do have neurotransmitter that's released and starts to diffuse outside of that specific region, you do have transporters that are able to uptake that excess neurotransmitter. Neurotransmitter diffuses out of the synapses and taken up via transporters. This image here that you can see is actually the excitatory amino acid transporter for glutamate. So this is an EAT transporter. You can see the the transmembrane domains. Again, this is going to form a pore that brings glutamate back into the cell. This image here I like just because it shows you kind of the different expression patterns of those transporters. So you can see that your postsynaptic receptors are represented kind of as these yellow and kind of, I guess, orangish colored, I don't know, shapes. But you also have neuronal transporters expressed here on your postsynaptic cell as well as your presynaptic cell. And then you also see that there are glial transporters that are expressed on the astroglial cell. So Sarah asked, since the sympathetic nervous system uses mostly G protein coupled receptors at the target organ, how does it cause a fast response? So when we say fast, I think that's kind of a very relative term. When we're talking, like, we're not talking like upon, I guess, milliseconds, which is kind of the I guess the timing in which ionotropic receptors are going to function. You see them opening and closing within milliseconds. G protein coupled receptors. So again, it's a time scale that I think is fast. When we say fast, it's very fast. And when I say slow, it's actually not really that slow, if that makes sense. You're still going to get a fairly instantaneous response, but it's not going to function as fast as, let's say, one neuron talking to another neuron. You think essentially the thoughts that are blowing through your brain right now are moving at a speed of like 200, I think, and 60 miles per hour. And so again, that's a very, very fast speed. So when I say slow neurotransmission, it's just, I think, a relative term to the fast. So you can still get a pretty instantaneous response with a G protein coupled receptor, but it's not going to be as instantaneous as with an ionotropic. Keith, would you agree with that statement?
Yeah, I think you're using the term relative is the way to go.
Yeah.
And when we talk about slow, like hormonal action versus neurotransmitter, the nuclear receptors, may take minutes or even hours to fully express an action. Whereas, like you said, here, even though the electrophysiologists have this terminology of fast and so on, the metabotropic is still fast in the context of the physiology.
Yeah.
Relative differences. Yeah. Thank you.
Yeah. No, and I always like hearing your opinion on this matter too. Thank you. Okay, so actually this is a good place to kind of pause for a second. So we just finished talking about transporters. So my concept question here is, what class of transport blocking drugs revolutionized psychiatric care? They were originally characterized in the 1980s. I think 1987 is what I saw, but they really probably took hold and became, I think, part of the culture in the 1990s. And so I'm just gonna, instead of sending you to breakout rooms this time, what I'm gonna do is I'm gonna have you go ahead and private message me your responses. So as soon as you kind of think you have the answer here, feel free to just kind of send me a PM. Okay, I guess the jig is up because everybody's like, I tried to PM, it didn't work. But yeah, so for those of you who have responded, thank you so much. I think everybody's kind of coming up with the right answer here, which is the SSRIs. Originally kind of characterized in 1987, SSRIs are going to be the selective serotonin reuptake inhibitors. Those inhibit the reuptake of serotonin, which causes extended, prolonged activation of serotonin in the synaptic cleft and reactivation of those receptors. When we get to the section of neurotransmitters talking about serotonin, we will no doubt discuss this a little bit more mechanistically, but at kind of, again, that 30,000 foot view, I just wanted to kind of bring in a concept that would show how transporters are, an important pharmacological tool that we use. Okay, so we have covered ion channels, receptors, transporters, and now we're going to start neurotransmitters, and this is going to be another big bulk of the section of this. Again, if you're following along through Goodman and Gilman, all of this is coming directly from there, and so we're going to start going through specific neurotransmitter systems. So when we talk about the neurotransmitters, probably the best way to start is by defining exactly how do you characterize what a neurotransmitter is, right? Maybe something falls outside kind of this idea of what a neurotransmitter is. And so from both textbooks, this is literally kind of the points that they give in order for something to be considered a neurotransmitter. So you can see Kazung has a kind of an abbreviated, but if you read these two structures, they're very similar and overlap. So we'll just go through them very quickly. So the transmitter must be present in the presynaptic terminal of the synapse and the neuron and in the neurons from which the presynaptic terminals arise. So there has to be some type of transmitter in that presynaptic terminal. The transmitter must be released from the presynaptic nerve concomitantly with nerve activity in high enough quantity to have an effect. So you have to have it in the presynapse. You actually have to have it be released when an action potential occurs. The effect of experimental application of a putative transmitter should mimic the effects of stimulating the presynaptic pathway. So if you put on some type of exogenous transmitter, you should still kind of have the same effect as if you had stimulated that presynaptic side. If available, a specific pharmacological agonist and antagonist should stimulate and block the measured functions of the putative transmitter. This is like awesome deep pharmacology, and honestly, I think I've gotten reviews from papers that have asked me to do this very same thing where you're saying this particular neurotransmitter is responsible for this function, and they ask you to both block it and mimic its action with some type of known pharmacological agent that has a high affinity for that receptor system. And then there should be a mechanism present, either reuptake or enzymatic degradation that terminates the action of the neurotransmitter. Again, you're going through almost the full cycle of the neurotransmitter at this point. You're proving that it's in the presynaptic area, it's diffusing across the synaptic cleft, that it's activating a postsynaptic receptor, that you can essentially mimic or block that action with other pharmacological agents that are known to have high affinity for that target, hypothesized target. And then lastly, there needs to be some type of termination of signal, whether it be through enzymatic degradation, as we saw with the acetylcholinesterases, or through reuptake, as we'll see through the EAT system or the DAT system, or a plethora of other transport systems that are available in the brain. And Kazung, again, the 3 points that they have here are very much overlapping with what we just went over here. Localization should be in the same area. Release, that should be released when you stimulate that neuron. And synaptic mimicry, again, you should be able to pharmacologically characterize that area. So this is how, if you were going to go propose that a new neurotransmitter system is in the brain, these are essentially the types of research steps you would have to take in order to kind of prove your point. Okay, so the neurotransmitter table. Ooh, I put this in here and I'm not quite sure if I'm going to be able to. Okay, so if you're going through this particular PowerPoint, this table is actually the table of all the neurotransmitters that we're going to be going over in a single table. I think it probably will link you to one of our pharmacology texts, and I just like it because it's a great study resource. It'll tell you the transmitter, block, antagonist, and the downstream kind of activating systems. And so when you get down to the point of studying, this table is going to be really useful and relevant, but we're going to see we'll see a version of these tables later on in this particular presentation. Okay, so in terms of neurotransmitters, the first that we're going to discuss are the amino acid neurotransmitters, and I don't know, maybe I'm I'm not impartial on this, just because I did study glutamate, but glutamate is the major neurotransmitter in the brain. So every thought that you're having right now is being mediated by glutamate, glutamate release, glutamate binding postsynaptic glutamate receptors. And so again, glutamate is kind of a big deal. Here you can see the structure of glutamic acid, as well as the the specific agonists that characterize the receptor subtypes. So NMDA, AMPA, as well as canic acid, and we'll talk about those specific subtypes here in a few slides. To complement the primary excitatory neurotransmitter, we have GABA and glycine, and those are going to be your major inhibitory neurotransmitters. These are neutral amino acids that are released by interneurons, and so the regions in which they're released are slightly different. GABA is going to be in your CNS and spinal cord, and glycine is going to be in spinal cord and brainstem. So I put these two things on this slide just because I think it's really important to recognize it's almost kind of like the gas and the brake pedal for the brain, and then all the other kind of systems are modulatory systems, not to downplay what they do, but these are kind of the major signaling systems that we see in the brain. And again, you can see the wonderful chemistry here, things like GABA, showing you some GABA agonists, as well as some GABA-A antagonists. So muscimol would be your GABA-A agonist, bicuculin would be your antagonist and picotoxin, and then your GABA-B would be baclofen and saclofen. And we'll talk about the difference between GABA-A and GABA-B receptors, again, kind of pairing off that ionotropic and metabotropic types of receptors that exist in certain neurotransmitter systems. Glycine, very similarly, you see here the structure, as well as glycine receptor agonists, things like taurine and alanine. Oh, and I just wanted to point out here at the very top of these slides, you can see that I've put things like central neurotransmitters and identification of neurotransmitters. Those are kind of the subsections of the textbooks in which you can find these materials in case you're looking for them. Okay, so the glutamate cycle. So again, glutamate is a primary excitatory neurotransmitter. So quickly, let's just kind of go through the cycle of how glutamate is synthesized, broken down, and released. So let's first start with release from the presynaptic cell via calcium-mediated endocytosis. So here you've got glutamate being released to the synaptic cleft, and it can bind to either ionotropic AMPA receptors, ionotropic NMDA receptors, or metabotropic mGluRs. So it's going to act on those postsynaptic receptors. Some of those receptors are expressed on the postsynaptic cell. Some of them are expressed perisynaptically, and there are some presynaptic glutamate receptors, although they're not represented on this figure. They do exist and have been known to generate some modulation in synaptic plasticity. Once it's released into the synaptic cleft, it's cleared by the glutamate transporters, the excitatory amino acid transporters, or EATs. There are 5 subtypes of EATs. EAT 1, 2, 3, 4, 5. EAT 2 is expressed primarily on glial cells. EAT 3 is expressed on postsynaptic cells. In glial cells, once it's taken up into glial cells through EAT 2, you have glutamate brought in. It is broken down from glutamate to glutamine via glutamine synthesis, and then glutamine is released and taken up into the presynaptic compartment. In the presynaptic compartment, through the enzyme glutamase, it's converted back into glutamate, and then it is transported into synaptic vesicles via V-GLUT. So you can see that there are several different places in which transporters play a role in this, and that it's interesting that in the glutamate cycle you have, once it gets taken up into those glial cells, it's converted into kind of an intermediary structure that's taken up into the presynaptic side, and then it's kind of almost like reactivated into glutamate and brought into those presynaptic vesicles. Okay, I'm just checking the time. Okay, so like I said, glutamate is a primary excitatory neurotransmitter. It can act through ionotropic receptors or metabotropic. So again, using those terms, fast and slow neurotransmission relatively. The ionotropic receptors, things like NMDA, AMPA, and kainate, and we'll talk more specifically about what ions are passing through those receptors. And the metabotropic receptors of the mGluRs. And so the mGluRs, metabotropic glutamate receptors, there are 8 different subtypes of those, and they're going to be coupled to different G proteins and thus have different downstream effects. Okay, does anybody have any questions? Again, I think a lot of this is review for most of the class, but I want to make sure that I answer any questions if they're popping up. So please feel free to populate the chat if you do have questions. Okay, so if we focus more on these subtypes of different glutamate receptors, here you can see the NMDA, AMPA, and kainate receptors as part of the ionotropic as well as, not surprisingly, the agonists for these receptors are going to be the original chemical that was used to characterize them, and then the most common antagonist for these. And so one of the most common antagonists for NMDA is going to be this molecule called AP5. For AMPA and kainate, it's CNQX. MBQX is a slightly higher affinity than CNQX for the AMPA receptor, and CNQX can also be used to block kainate receptors as well. So to answer your question, is there a reason that glutamine and glutamate need to be interconverted in this process? If you think about glutamate as a signaling molecule, if once it goes into the glial cell and it is transferred from the glial cell back to the presynaptic cell, you want to make sure that it's not activating glutamate receptors on that presynaptic cell. And so using that other molecule, that interconverted the glutamine, it allows it to kind of move between that synaptic area without activating glutamate receptors. And so you don't want to cause more glutamate to be released non-specifically in that region. Does that make sense? So it kind of makes it I don't want to say lame duck, but yeah, it makes it so it's just not activating the receptors in that region unintentionally, non-specific activation, which would possibly cause exocytosis or excitotoxicity, which we'll talk about later. Okay, so again, this the function of this table again is to just show you the different families of receptors, the different subtypes that are going to comprise these. We'll talk about how these different subtypes are composed within the receptor in following slides, and then the agonist and antagonist. As an electrophysiologist and I guess kind of a pharmacologist, I would say that this table is super helpful, because if I was studying, let's say, the NMDA receptor system and I wanted to block it, I could come to this table and say, okay, I just need to buy some AP5 and make sure I apply it to my brain slice. This is, I guess, kind of a version, the same table that we just saw, a different version of it though. I think I'm going to continue to add to this table, because what it basically is a converted version of that larger table that I mentioned at the beginning of this neurotransmitter section, because all of the different neurotransmitters are kind of in this particular table structure. So every time I talk about a different neurotransmitter, I'll show you this version of the table and we can kind of continue to add on to it. Again, if you're a person who studies via table, they can be really helpful, because they allow you to kind of compare and contrast all of these different things that you're learning. And although it might seem like as you're being presented the information that the differences are subtle, I think once sometimes you see it in table form, it becomes a little bit more apparent. Okay, so the different types of ionotropic receptors. So the NMDA receptor and methyldiaspartate receptor. In terms of the way that it's composed, it's a heteromer, and it's going to be composed of at least a gluN1 and a gluN2 dimer. That's the minimal composition for an NMDA receptor to be functional. And it's highly permeable to calcium. Now, it's the influx of calcium that makes NMDA receptors very important for synaptic plasticity, for memories to form within your brain, for the process of long-term potentiation to happen. NMDA receptors have to be activated, and this is biology, so you probably could go out there and find a paper that says NMDA receptor independent long-term potentiation. I'm sure that paper does exist, but for all intents and purposes in textbooks, NMDA receptors activation is important for long-term potentiation or potentiation of synapses to come together. And we'll talk about that more when we talk about the process of synaptic transmission, but just recognize the importance of NMDA receptors in that. And again, it's the influx of calcium that mediates that signaling process and causes some of the downstream signaling cascades that causes the long-term kind of increased potentiation between two synapses and causes the brain cells that fire together to wire together. We're going to talk Hebbian. So these NMDA receptors are blocked by magnesium. They're voltage dependent. They're blocked by magnesium. Again, if you think about it, magnesium is a positive cation. It's kind of blocking the hole in that pore. This magnesium block actually makes NMDA receptors coincidence detectors, meaning that two things have to happen in order for an NMDA receptor to be activated. You have to have the ligand binding to the NMDA receptor. That's the first thing that has to happen. But the second thing that has to happen is you have to have depolarization of the cell. So you have to have the cell depolarize enough, become positive enough that it pops out that magnesium block. And so you have to, so it basically, it detects two things at one time, right? Depolarization of the cell, as well as the presence of glutamate. And so if both of those things are happening, it tells the cell essentially that, you know, glutamate is really here. We're really signaling. This might be an important event. And so NMDA receptors are considered coincidence detectors, the coincidence of those two events. They also require glycine to bind, but glycine is pretty ubiquitous in the brain and is important for synaptic plasticity, as I kind of talked about there for a moment, the role of it in long-term potentiation, which is the molecular mechanism for learning and memory, which we can talk about at length if you like. Love LTP. So NMDA has very slow kinetics. The very fast kind of kinetics of the brain occur. And again, these are both ionotropic, so I use that term very relatively slow and fast. AMPA receptors have fast kinetics. There are heterotetramers, but they're permeable to both sodium and potassium, but not calcium, so they don't have that same type of role in plasticity because you have, they're not permeable to calcium. They have fast receptor kinetics, so they open very quickly. They also desensitize very quickly, and so as quickly as they open, they'll close. If you puff glutamate onto a brain cell or a single cell and you look at the excitatory response, you're going to see a very fast peak with your AMPA receptors. And if you isolate just your NMDA receptor response, it's going to be kind of like a slower, longer type of response. But those AMPA receptors desensitize very, very quickly, kind of stopping the rest of that response. But they also are responsible for synaptic plasticity because they assist the NMDA receptors in being activated because they are permeable to sodium. So they cause that postsynaptic depolarization that aids in the relief of that magnesium block. You can get AMPA receptor independent LTP though. Catenic acid, lastly, a hetero or a heterotetramer requires extracellular sodium or chloride. Does not exist in the postsynaptic signaling complex and does modulate excitability. Okay, so if we look at the NMDA receptor, this figure does a great job of showing all of the different binding sites for different drugs. So you can see here the glycine binding site on different agonists and antagonists. So your agonists are shown in green, your antagonists are going to be shown in red. So you can see different, the glycine site is also binds D-serine, glycine, polyamine site, the PCP site, which can also be antagonized by MK801. The glutamate site obviously is agonized by NMDA. You can see here also the magnesium binding site. Zinc can also act as an inhibitor for NMDA signaling because of its divalent cation status. And then you can also see here on the intracellular side, there are regions in which the NMDA receptor can be modulated by kinases. And so there are ways in which this NMDA receptor signaling can be modified in that regard. What's nice to also recognize from this particular figure is that the NMDA receptor is permeable to both sodium and calcium. Okay, questions or concerns? Okay. Okay, so because of, like I said, the importance and the role of calcium, calcium homeostasis is important to keep tightly regulated. If it is dysregulated, it can lead to things like cell death. So during excess release of glutamate that can occur during ischemic events or in hypoglycemia, you have large amounts of glutamate that get released. This can be due to release from either neurons or from glial cells, but that release of glutamate is going to activate both mGluRs as well as the ionotropic receptors. And the activation of those different types of receptors is going to lead to large amounts of influx or large influxes of calcium into the cell. You can see how, obviously, because the NMDA receptor is permeable to calcium, how it comes into the cell here. With glutamate release and you're activating, let's say, the AMPA receptor, there are voltage-gated channels that are going to be opening because of the depolarization that are permeable to calcium as well. So you're also going to see calcium influx through those voltage gated channels. It's going to exacerbate the influx of calcium. The mGlu-R activation is going to, again, in a relative way, be a little slower, but the coupling to the G proteins is going to lead to release of intracellular calcium from the endoplasmic reticulum. So overall, kind of the big event here is the influx of calcium leads to possible excitotoxicity, coupled, which eventually will lead to apoptosis within the cell. So again, this is one of the ways in which cell death occurs during an ischemic event. And because of that, this whole process, some of these particular receptors have become targets for therapeutic design. So if we can possibly mitigate the influx of calcium during an ischemic event, possibly we can stop this whole pathway from occurring within those cells. Okay, so we've talked about the ionotropic receptors, NMDA, AMPA, and the kainate receptors. So kind of, those are all the ionotropic receptors. We also have the metabotropic receptors. So these G protein coupled receptors, they have 7 transmembrane domains, like all G protein coupled receptors do. There are 8 different subtypes, and they're grouped into 3 different kind of categories. Those 3 different groups are kind of grouped via their G protein coupling and their downstream effects. You can also kind of see here the distribution of those metabotropic receptors as well. So here you see, if we look at a postsynaptic cell, you have your mGlu-R 1s and 5s, mGlu-R 2s and 3s. If we look at a presynaptic glutamatergic cell, you see mGlu-R 2,3, mGlu-R 7, mGlu-R 4,8, mGlu-R 1,5. If you look at the presynaptic GABAergic, so again, the cell is different from this one because it is GABAergic, it is an interneuron. You can see that there are, cut it off, there are mGlu-Rs, this is mGlu-R 7, and then mGlu-R 2,3. And then if we look on the glial cells, you can also see that there are mGlu-Rs expressed on those glial cells. Group 1 mGlu-Rs are going to activate phospholipase C and cause intracellular calcium release, as we saw in the previous slide with excitotoxicity. And group 2 are going to suppress excitability because they're coupled to the GIO, inhibit calcium channel, or inhibit the calcium channels. And then this should say group 3, sorry, that's a misnomer there. This also suppresses excitability and inhibits presynaptic release of calcium. So again, the 8 different subtypes of the mGlu-Rs. If we go back a few slides, this is also a really nice table for that purpose. You can see the different groups of metabotropic receptors and which subtypes are included in which groups, and also some of the downstream signaling effects in the G proteins are coupled to. So if you have a hard time kind of with the grouping structure, this table can be really advantageous in that way. Okay, so we have about 2 minutes left, so this is going to be, I think, the last thing that we do for today. So the drug, Racetam, is not approved by the FDA, but is sold in Europe. What is the target of this drug, and what effect does it have on its target, and what is the proposed use of this drug? So I'm going to do a quick, what does this drug do? So I'm going to let you do this individually. I'm not going to send you to breakout rooms, so go ahead and use the Google or whatever web browser you like, a search engine, to do a quick search on this particular drug. And I want to go ahead and send me a private message when you come up with your answer here about this particular drug. What does it do? What does it do? What is its target? What does it do? And then what kind of larger effect on the body is this drug supposed to have? Okay, so somebody's come up with the correct kind of application of this drug, what is it for? Can anybody find, here we go. Okay, so great. I think you guys have stumbled upon it. So what this particular drug does is it positively modulates AMPA receptors. It actually blocks desensitization of AMPA receptors. And because of that, it's used as a nootropic, meaning that it improves cognitive function. So if you think about those AMPA receptors in the synapse undergoing desensitization, it's kind of shutting them off. This particular drug is used in Europe as a nootropic to improve cognitive function. But it's also shown to do a series of other things, alleviate anxiety and treating a variety of other neurodegenerative disorders. But it does act as I don't know if I'd necessarily call it a positive a PAM, but it does block desensitization of AMPA receptors. Okay, so really quickly, as you guys are kind of in the chatting, I guess, mode, do you guys like or hate the concept checks? Again, it's just kind of a way to break up lecture and kind of hopefully re-engage you with the material. So let's get either a like or hate. Okay. Just want to make sure. Okay, you don't like the breakout rooms. Good to know. Okay. Okay, cool. Yeah, just want to make sure that what I'll try to do, I know the breakout rooms, they do slow things down to a considerable amount, but they do allow you to kind of chat with each other, which I like. So I'll try to kind of keep the breakout room use to a minimum and then probably use the chat a little bit more and even the polls if I have some multiple choice questions. Okay. Well, that's all I have for you, everyone, today. Thank you all for your attention. If you have any questions or concerns, please feel free to reach out to myself or Keith. And Keith, do you have any questions or not questions? Do you have anything to add?
Just one little comment. If we go back to that interconversion of glutamate to glutamine and back to glutamate, we have to keep in mind that these are amino acids and that they have functions beyond neurotransmission, which normal amino acids have. And another aspect of the glutamate-glutamine deal is I think it actually links up with the urea cycle. And there's a relationship between nitrogen metabolism and this concept. So just thought I'd throw that complication in there.
No, I think it's a great point. And then as we talk about GABA, we'll also see kind of how that also plays a role in like the cycle and the use of the glutamate as a precursor.
Great. Thanks, Katie. Very interesting.
Yeah, no, thank you so much. And thank you everyone for your attention and have a great day.
Hey, Katie, I have a question.
Yeah.
So like these slides are super in depth and like a lot of information in just the 2 classes that we've had. Yeah. So when we're going back and like reviewing these central nervous system slides, like how would you recommend us pulling out like the most, like, do we just want to get like the broad concepts, or should we more focus on even the smaller details, or is it the smaller details that are what are important?
I think right now, like we're going over a lot of broad concepts that are going to be applied to like the specific neurotransmitter systems, and that I think was where we'll see more details. In every other class that I've ever done, I've always created study guides for students because I think people generally find them very helpful.
Okay. To kind of help narrow down
focus. I'm going to talk with Dr. Parker to see if he thinks that would be amendable in this class. And if so, then I might be inclined to do so. So yeah, so there might be a study guide coming your way, which I think would help focus your studying as well.
Okay, perfect. Okay, yeah, because I was going through these and I was just like, man, it's just a lot of information. And a lot of it is review, but okay, perfect. That answers my question.
Okay, thanks.
Thank you.